Okay, moving on to the next topic, we will be having the recombinant DNA technology. Though we already discussed this topic before, now we are going to discuss it in more details. So recombinant DNA technology joining together of DNA molecules from two different species. And why do we exactly need it? So we produce insulin, growth hormone, vaccines, enzymes, antibiotics, gene therapy, and so on. So how exactly it happens? I'm just gonna give a short summary. First, we find gene of interest like insulin gene or growth hormone. Then we choose a vector. Vector carries the gene into another cell. The most common vectors we can have plasmid, virus, or bacteriophage, though plasmid is the most common. So the small circular DNA is easy to modify. That's why plasmid is the most common, but virus and bacteriophage can be used as vectors as well. Then we cut the DNA using the same DNA of gene and DNA of plasmid using the same restriction enzymes so that we can produce sticky ends, and they make it easy to pair. Then we have DNA ligase. It's the ends they stick, but DNA ligase glues them together like final glue. Though they naturally stick, they can later become weaker. Then we have transformation. It's the uptake of foreign DNA by bacteria. And then we have selection. So what is selection? Like, how do we know which bacteria receive the plasmid? So this is our question. And we have the antibiotic resistance gene. Basically, when we apply these genes, the bacteria with the inserted plasmid, they survive. So we have, and then after we have selected them, we do cloning and protein production. So what is cDNA? cDNA stands for complementary DNA. And what is the problem? Human DNA has exons and introns, and bacteria cannot remove introns. Bacteria doesn't understand it. So if we simply use the DNA, the introns are going to be copied in the final result as well, which is not what we want. So as we discussed in the this molecular biology central dogma, so DNA, we have to RNA, it turns into RNA and it turns into protein, right? But we can take this mRNA that usually helps to produce proteins, and we turn it into we go back, we turn it into the DNA, but it's called complementary DNA. And in the mRNA already the introns, they have been removed. So we basically do not work with introns if we apply the reverse transcriptase process. So instead of genomic DNA, scientists use mRNA. It's like we take mRNA and we turn them into complementary DNA. And now we have solved the problem, and it contains exons only, no introns. Let's talk about polymerase chain reaction, which stands for PCR. It's basically how to make millions of copies of DNA. It consists of 3 steps. We have denaturation, annealing, and extension. In denaturation, we heat the DNA and double strand they separates. In annealing, we cool the DNA and primers they attach. And in extension, as we do it many cycles, usually 25 to 30 cycles are done, and it can produce millions of DNA. That is amazing. So I would like to show you more about PCR, polymerase chain reaction. It's a method used to make millions of copies of a specific DNA sequence. And it also helps scientists detect small amounts of DNA in samples. So How exactly it happens? There's 3 steps of PCR. First is denaturation. We apply heat so that the 2 strands they separate. Then we apply annealing. As you can see the temperature change here. The primers they attach or bind to the target sequence on each DNA strand. The primers they are going to attach on the target sequence. And then in extension, DNA polymerase builds new DNA strands from the primer. So we just let the DNA polymerase do its job. And it builds the new DNA strands from the primers that has been attached. And when we do 25 to 40 cycles of this process, we can have the millions of copies. You can see cycle 1, 2 copies, 4 copies, 8 copies, and suddenly we can have millions of copies. As the number of DNA copies increases exponentially. Okay, so then we have a thing called agarose gel electrophoresis. It is basically how to separate DNA fragments according to size. That's what we need in recombinant technology as well. So DNA is actually negatively charged, and electric current is applied for the DNA to move toward the positive electrode. And then what happens? We prepare agarose gel. It acts as a sieve. Small DNA fragments in this agarose gel, they move faster and farther. The large ones, they move slower and stay near the walls. And you can see we have a different bands at the end. We get it as a result. So it checks DNA size, restriction, digestion, PCR products, and if insert is present. So now let's discuss a vector. What are the good characteristics of a good vector? So it replicates independently. It has multiple cloning sites, have selectable markers. We are going to talk more about it. It does not degrade. So we have 3 types of vectors: plasmid, cloning vector, virus chromosome. Plasmids can be supercoiled, open circular, and linear. So best plasmid size for cloning is up to 10 kb. And let's classify the plasmids. We have fertility plasmids, helps for this conjugation. Resistant plasmid, antibiotic resistance. Cold plasmid helps with toxins against other bacteria. Virulence plasmid, it's a pathogenic one, causes pathogenicity. Degradative plasmid allows bacteria to break unusual chemicals like toluene or salicylic acid. Let's talk about conjugation, about plasmid transfer. So we have donor cell, pilus forms, then it moves to recipient cell, and plasmid is transferred. So I just want to show you quickly. So plasmid conjugation. Okay, so you see here, just we have donor, the pilus forms, connects to recipient, then DNA polymerase helps it to move, to transfer, and then each of them, we have got old donor and new donor, and they all both have plasmids. Anyways, so now I want to talk about PBR322. It is one of the famous plasmids, and it's got 3 important parts. First, it has origin of full replication. Let me see if I can show you. PBR322. Yeah, this is the PBR322 plasmid, and it's quite famous, as you can see, quite famous. And it has got origin of replication, where plasmid replication starts, it has ampicillin resistance genes, and it has tetracycline resistance genes. You see here AMP and TET. Interesting, right? So why it has two antibiotic genes? If we grow on ampicillin, it lives. And if we grow on tetracycline, it dies. And if these both conditions are present, it means that the gene was successfully inserted. So we grow it on agar, and if we grow it on ampicillin and it lives, and later we grow it on tetracycline, it dies, it means the gene was successfully inserted. Then we have insertional inactivation. So what is that? Its inserted gene disrupts a marker gene, causing loss of a function. So restriction map shows where should we cut the plasmid. So then we have, so I hope you understood what it means. Insertional inactivation is when inserted gene disrupts a marker gene, causes loss of function. And so then we have PUC8. So we have discussed the PBR322, but later scientists made better plasmid. So let me just show you. PUC8. Yeah, this is the, I would say, improved one. So it's smaller, it has got more copies, easier cloning process, and better selection. So we do not need to grow it on ampicillin or tetracycline like before. Now what we do exactly? We have a zinc, as you can see here, we have LAC-Z, right? So we have LAC selection. Basically, we can describe it as blue-white screening. So instead of antibiotics, scientists, they use colors. So if it's normal bacteria, it gives us blue colonies. And if foreign gene has been inserted, the LAC-Z gene is disrupted. So colonies, they become white. So blue, normal, white, plasmid, present. We can remember it like that. So if we compare them, PBR322 versus PUC8, we have two antibiotic markers, and here we have one antibiotic marker, ampicillin resistance, but here we have ampicillin and tetracycline, plus just like Z. Selection by antibiotic resistance, blue-white screening, larger plasmid, smaller plasmid, lower copy number, higher copy number. Copy number basically is how many plasmids it contains. Now let's talk about X-gal. X-gal is colorless chemical, and some bacteria produce an enzyme called B-galactodiase. And this enzyme actually cuts the X-gal. Let me show you. X-gal and beta-galactodiase. So which one would be suitable to show me? Okay, let's say this one. We have X-gal staining, and you can see the difference. Positive white, negative blue. You can see that. And we have mentioned here, blue is normal, white is plasmid present. So basically, X-gal is a colorless chemical, and some bacteria produce an enzyme beta-galactodiase, and this cuts X-gal. And when they're combined, we have blue color. And PUC8 contains many different restriction enzyme sites, and LAC-Z gene makes beta-galactodiase. Okay, and Let's talk about directional cloning. Two different restriction enzymes, so the gene inserts in one direction only. So in this case, we apply two different restriction enzymes, and the gene is going to be inserted in one direction only. Now, so how do we use recombinant DNA exactly in a lab? If we take a useful gene and we make bacteria copy it, that's basically what we gotta do. So first we obtain the gene, we cut it, we cut the plasmid gene, we join them together, insert into bacteria, select transformed bacteria, identify recombinant, confirm gene presence, and produce proteins. So what are our sources of genes? Can be plant, animal, microorganism, synthetic DNA. Gene preparation. Gene is isolated by PCR or restriction digestion. So this is our gene preparation, but we also need to have vector preparation. Cloning vector is isolated and cut with restriction enzyme, and then we have insertion, or we call it ligation. It's when the gene of interest is inserted into vector using DNA ligase to form recombinant. Ligase sealed DNA and recombinant plasmid is formed. Then we transform it into host. Selection and screening. Transformed cells are growing on selective media. Colonies containing recombinant plasmid are confirmed. Then we have expression of genes, and then we have product recovery and purification from culture, and then we have product of interest for chemical engineering. So example products, we have got lipase enzyme, we have biodiesel production, like let's say lipase enzyme can give us biodiesel production, ester synthesis, lipid modification, surfactant and flavor production, and so on. These are just examples. Now let's talk about nucleases. Nucleases are enzymes that cut nucleic acids. They break down phosphodiester bonds. We have endonuclease, which cuts from the middle, and exonuclease, cuts from the end. They are used in recombinant DNA technology. Restriction enzymes are endonucleases. Exonuclease cuts from the end, removes nucleotides one by one. It is responsible for proofreading and DNA repair. And endonuclease cuts inside of DNA, makes internal cuts, and it's responsible for recombinant DNA technology. So what is restriction endonuclease exactly? Recognize specific DNA sequences and cut them. We can have recognizing specific DNA sequence, for example, hexanucleotide sequence containing 6 DNA bases, and most recognize hexanucleotide targets, most of them. So most are going to recognize when we have 6 DNA bases, but some they can cut at 4, 5, or 8, and so on. And then we have degenerate recognition sequence. Basically, like if we have like it's N, it means any nucleotide. Enzyme recognize all of this as family of related sequence. So let's talk about 3 types of restriction endonucleases. So we have type 2 restriction, polymerases, and ligases. Type 2 restriction cuts DNA by breaking phosphodiester bonds. Polymerase builds new DNA, ligase joins DNA pieces. And then we have sticky ends versus blunt ends. If we cut it unevenly, the single-stranded overhangs, then we have sticky ends. So they are easy, base pair, easy to join, preferred cloning. For example, exori. But we also have when we have straight cuts. The pros are no overhangs, cons are harder to join. It's used when there is no choice. So it's like an exception. Polymerase synthesizes new DNA strand using an existing DNA as template. Adds nucleotides, makes complementary DNA replication, PCR. Then we have ligase, joins DNA fragments by creating phosphodiester glue, phosphodiester bonds. It's like DNA glue. Repairs DNA breaks, joins DNA fragments, produces recombinant DNA, joins 3-OH with 5-phosphate. These endonucleases work together. So first we have restriction enzyme cuts DNA, and we have sticky or blunt ends. Sticky ones may pair. And then we have DNA ligase, glues them together, new phosphodiester bonds, and polymerase copies DNA. So then, how do we introduce it into living cells? First, we have the process of transformation, plasmid to bacteria. And then we have identification of recombinants. But we have got an issue. Bacteria don't want to take foreign DNA inside of it. So DNA is negative because of its phosphate groups. And bacterial cell membrane is also negatively charged, so they repel each other. So what we do, we treat bacteria with calcium chloride. Calcium has positive charge. They neutralize negative charges on DNA bacterial surface. They allow plasmid to get closer to cell membrane. So then we get competent cells, the ones that are capable of taking up foreign DNA. And then after calcium treatment, plasmid attached near the cell surface. Plasmid is attached near the cell surface. And then suddenly we raise the temperature, so heat shock makes plasmid DNA to slip inside. After heat shock, bacteria are stressed. They need to repair membrane damage and recover from heat shock, and they start expressing resistance genes. So entire process, first we treat bacteria with calcium chloride and form competent cells. Then we add plasmid DNA, plasmid attached to bacterial surface. Then we perform heat shock. Temporary pores form, plasmid enters cells. Then we have recovery, antibiotic resistant gene begins working. And then we place it on antibiotic agar, and only transformed bacteria, they survive. So how to select for transformed cells? We use selectable markers. Example, as we've mentioned before, in PBR322, ampicillin resistance gene and tetracycline resistance gene are the selectable markers. Let's say we have transformed cell and non-transformed cell, and we place them to grow. The transformed cell will not grow anything, but the ampicillin resistant colonies, they are going to grow. So we want recombinant plasmid, not self-ligated plasmid. So we can either get recombinant plasmid or we can get self-ligated plasmids. Plasmid closes back on itself without inserting gene. That is a ligated plasmid. So gene is not inserted and plasmid closes back on itself. So plasmid and nothing. But in recombinant plasmid, we have plasmid and gene of interest combined. So our solution is insertional inactivation. Basically, inserted DNA knocks out or inactivates a marker gene. How does it happen exactly? The method is to identify recombinant bacteria by inserting foreign DNA into marker gene and disrupt its function. It's a method. So we identify recombinant bacteria by inserting foreign DNA into marker gene and disrupting its function. Let's say we have a PBR322 and gene inserted into tetracycline. If it's a recombinant, ampicillin resistant works, but tetracycline is broken because we have had a gene inserted there. But if it's self-ligated, the both are going to work because anything is going to work. It is just a plasmid. It has not contains a gene of interest. And let's talk about PUC plasmids. They have got a high copy number. One bacterium gives us many plasmids. They have got blue-white screening. They contain LAC-Z gene and uses X galactase to confirm the presence of recombinant gene. So let's just do the final review. So we have polymerase chain reaction, amplifies specific gene region, presence of band of expected size. Then we have gene for electrophoresis after PCR and restriction digestion, visualizes DNA fragments. Then we have restriction enzyme analysis, cuts DNA with specific enzymes to confirm presence and structure of gene. Then we have Southern blotting, detects specific DNA sequence in the sample. Then we have colony PCR. If we use bacterial colonies, quickly screens colonies for presence of gene insert. And then we have DNA sequences, provides exact sequence of genes. These are all the methods to confirm the presence of recombinant gene. So we can use PCR, gel electrophoresis, restriction enzyme analysis, southern blotting, colony PCR, the DNA sequencing. To let's say we see the southern blotting steps. We extract DNA from sample, fragment DNA with restriction enzymes. We get fragment DNA with restriction enzymes. We separate DNA by size using gel electrophoresis. We transfer and fix separated DNA molecules from the gel onto nylon membrane. And we probe for the DNA of interest using a nucleotide probe. And radio-labeled probes can be analyzed after they're detecting using X-ray, film, or phosphor imaging using a laser scanner such as the new Azure and blah blah blah, like we see here. So, or we have colony PCR protocol. Pick colonies with sterile pipette. If using LaxG reporter, pick white colonies because it's going to the X-gal is going to cut it, you know. Beta galactodiase, which is present, is going to cut it. So we pick white colonies. Add colony to PCR tube, stir to resuspend. Run PCR with an extended cell lysis step and run DNA gal. Check for expected PCR products. So these are just some of the examples to confirm the presence of recombinant gene. So this is it about the recombinant DNA technology. Thanks for watching. Have a nice day.